My name is Andrew Buckler, and I have been asked to discuss the growing role of quantitative imaging in pharmaceutical discovery and development. I've spent 25 plus years working in the field of imaging instrumentation and applications, and presently own a consulting practice as well as an early-stage company working to develop and optimize quantitative imaging as a primary tool for drug development as well as patient management in the clinic. Until now, clinical radiology has been composed of a radiologist review of imagery being undertaken with a variety of conditions but interpreted essentially the same way, which is for the reviewer to visually evaluate the image, possibly with the use of simple screen tools like calipers for the measurement of line lengths. However, an increasing number of quantitative analysis approaches have been pursued that create objective and quantitative readouts, which range from morphological to functional measurements. Some of these methods use single images, while others use dynamic sequences that afford the assessment of time-dependent behavior. But in general, they're one form or another of numeric computation using a large amount of the available data from the image to provide a measure that is more closely related to the underlying biology than may be easily appreciated by the human eye. These capabilities are used preclinically, in translational medicine, and increasingly in the clinic as well. The FDA's Critical Path Initiative calls for modernizing drug development by incorporating relevant scientific advances such as genomics and advanced imaging technologies into the process. Early detection of drug response can significantly reduce the time required to conduct proof-of-concept studies for new drug candidates, and quantitative imaging may offer a sensitive means of defining the clinical dose range. If such an early effect can be shown to be a valid predictor of clinical outcome, quantitative imaging could serve as a surrogate endpoint in phase 3 trials. This would significantly reduce the time and cost required to make new treatments available. Advanced imaging technologies also provide solutions for diseases and therapies where conventional response assessment techniques have recognized limitations, such as targeted and personalized treatments. In general, quantitative imaging may be used to efficiently evaluate whether an experimental therapy is effective. In preclinical, it is possible to confirm activity in vivo and explore exposure-response relationships. For exploratory clinical, it is possible to establish the presence and coverage of the target, stratify study populations, and ultimately contribute to go/no-go decisions on whether to continue development. Likewise, as a phase 2 trial endpoint, quantitative imaging may be used for better patient selection, to provide an early assessment of response, and to predict which patients are at risk of recurrence. Quantitative imaging provides tomographic information with exquisite spatial, temporal, and biochemical resolution in vivo. Its non-invasive nature facilitates serial studies in animal models, as well as patient compliance in humans. Imaging phenotypes can be used to subclassify histologically similar tumors to be angiogenic, hypoxic, or proliferative subtypes, by way of example. The diagnostic confidence in assessing response depends on the measure and how much of the available data is utilized in order to determine it. As a simple but important illustration, tumor size assessed by longest diameter uses 2 points within a single image slice, whereas a volume measurement interrogates all 3D pixels in the tumor. After therapy, due to the irregular nature of tumor change, the line length and volume measurements potentially lead to different conclusions. Using all of the available pixel information allows the shape of the tumor and any heterogeneities in how it changes with time to be factored out and achieve a more accurate measure of what counts, namely whether the size has changed or not. Statistically, the fact that there is less noise introduced into the measurement, as well as the fact that it may be less prone to asymmetric error in the trial arms, allows the measurement to react more quickly to the real biological change. As a result, shorter trial durations and/or lower patient enrollment for a given level of statistical power is the driving motivation. New models for therapy development, in this case for cancer, are enabled by more sensitive measures that are tied closer to the underlying biological mechanism than previous methods. Use of quantitative imaging early allows better patient selection and more possibility to move patients across faster-paced programs to both serve individual patients as well as provide business results due to new technology options and reduced costs for standard trial designs. Another promising modality, arguably even closer to the underlying biology, is PET. FDG-PET is recognized as a sensitive tool for the diagnosis and staging of a number of solid tumor types and assessing their response to treatment. Cancers have an increased rate of glycolysis and glucose transport. 18-fluorine fluorodeoxyglycose, or FDG, serves as an analog of glucose and can be used as a marker of the metabolic activity of cancer. FDG is trapped intracellularly following ingestion, uptake, and phosphorylation. Changes in FDG uptake after treatment reflect the cellular response to therapy. FDG-PET can be used as a translational method because it is used in animal models as well as in humans. It is well established in a number of indications as contributing evidence in phase 3 trials, including Alzheimer's disease, stunned myocardium, lymphoma, infected joints, and others. Moving further into the capabilities of imaging, beyond the use of a single volumetric image and into the evaluation of a dynamic series of them, is possible with pharmacokinetic modeling. There are a rich set of modeling applications in PET, but I'll highlight a promising dynamic MR example. The power of MR lies in its exquisite spatial resolution, which allows it to quantitatively assess how and where contrast agent accumulates in and around neovasculature, such as is often considered a hallmark of cancer. Specifically, prior to forming complete vessels that recirculate well, there is a microvasculature experienced as permeability through tissue that can be measured in images as an enhanced region. This enhanced region may further be analyzed in terms of the time it takes for agent to build as well as to flush from the area, and serves as an appropriate measure for drug programs targeting anti-angiogenic effects. Like its counterparts in dynamic PET, this measure is arguably even more sensitive to drug action than the single image volume case that I discussed. If we look at where this is going, we observe that technology development has enabled increasingly targeted means to quantify pathogenesis. Quantitative imaging offers mineable phenotypic features that are objective readouts of tissue characteristics, capable of being used in numeric hypothesis testing. They cover a range of biological scales, from organ systems down to tissue characteristics in the microenvironment. These features may be extracted and mined along with other quantitative features from scales above and below to add significantly to the predictive power of pharmacogenomics. The diversity of imaging phenotypes has been demonstrated as being greater than that of histology in an increasing number of settings. Likewise, whereas it has been observed that the genetic predisposition of an individual has a large bearing on what diseases they're subject to, and others have argued that we have considerable amount of information that come from measuring the phenotypes, perhaps the most predictive power lies in the clever cross-correlation across these heterogeneous data, rather than relying only on one type alone. Whether in the full multi-scale context or in more limited applications, it is clear that biomarker development in clinical radiology now and moving forward into ever more sensitive and high-resolution imaging assays will provide increasing opportunity for new drug development programs and patient management. An initiative referred to as the Quantitative Imaging Biomarker Alliance, or QIBA, has been undertaken for the last few years by stakeholders across practicing clinicians, biopharma companies, imaging device manufacturers, regulatory and non-regulatory government agencies, and academicians to organize a collective effort to pursue the qualification of agreed markers. This effort has developed a procedural template and a process roadmap that is presently being applied in multiple projects. The outputs of the effort are profiles that are structured to address the scientific nature of biomarkers. The intention is that as users and suppliers work together on these profiles, that the necessary qualification converges, and with a device to accomplish the standardization needed for wide availability. In summary, I hope I've convinced you that beyond just diagnostics, images are non-invasive biomarkers at the frontier of measurement science, and that quantitative imaging biomarker characteristics meet key requirements for endpoints to increase the speed and reduce the cost of trials. As illustrations, volumetric CT seeks to increase sensitivity, and molecular imaging with PET and pharmacokinetic modeling with PET and DCE MRI measure closer to underlying pathogenesis, enabling new models for clinical trial designs. I've also defined quantitative imaging as the extraction of mineable phenotypic features inside the broader classification context needed for precision medicine. And I've highlighted Kiva as an organized activity for multi-stakeholder standardization to increase the pragmatic utility of this important technology. Thank you very much for your interest, and I wish you the best with your programs.